thanks a lot for being here. Thank you for the introduction. It's been, I'm looking forward to an interesting 3 days. So what Elliot asked me to do is introduce a little bit the question of tracking and predicting the spread of RNA viruses. And I'll mostly focus here on human seasonal influenza viruses, with this kind of real-time evolution, tracking of real-time evolution that probably has been going on for the longest. So just before I get into it, I consult for Moderator as a disclosure. Yes, human seasonal influenza viruses. So, these are only the influenza A viruses, lineages circulating in humans, and you probably all know those. They are, they are designated by these two-part acronyms: H1N1, H2N2, H3N2. This is a famous Spanish flu. This is sort of a flu that emerged in 57, and then H3N2, which is still circulating today, in 68. And this is a swine origin pandemic influenza 09. And I guess we'll hear more about this interface with swine later today from Tavis. So I'll mostly look into influenza H3N2, where sort of we have seen the kind of the most rapid evolution and the most long-term coverage of real-time evolution tracking, if you want, sort of mostly as an example and as sort of a use case where genomic classification, subspecies classification has been used and is being used in a sort of continual fashion through the vaccine updates that we get every year. I won't go into this classification here, which is sort of initially, I think, defined serologically, whether that is subspecies or minus species or any of the other variants of this. I leave it to others to decide. They typically referred to as lineages in the seasonal flu community. So this is, if you look at just sort of influenza case numbers, weekly positive tests in the US by the US CDC collated here. And I've just cut this off here up to the point where the 2009 pandemic started. And you see sort of a happy back and forth between the H1s and the H3s. Some years are dominated by one, some by the others. And then there's years where both of them seem to be circulating continuously. If I also put in the influenza Bs, they typically be a little bit shifted further into the spring and have a secondary peak there. So now in 2009, many of you will remember, suddenly, basically at the end of the flu season, there was this, in March, these reports of a novel H1 influenza, which of course, knowing that the Spanish flu was an H1, sort of caused a lot of concern. And this resulted in sort of an anomalous season with sort of a double peak, first in summer in the US, and then sort of a later peak in early flu season, basically in October, November, sort of a few months shifted. So it has sort of a bit like we've had this with COVID, sort of double peaks. It's sort of a bit unpredictable timing. Then there was one flu season which is basically absent. The old H1 was gone, the new H1 took its place, and they have been kind of continuing the same sort of dance that was going on before, only that the old H1 was replaced by the new H1. And then sort of the pandemic came, and we didn't see much of that for about a year and a half. Now everything's back to normal. So what does it take for such a virus to kind of to endemically circulate? Some way or another, it needs to find individuals to infect, right? It needs susceptible individuals, which could either happen because there's new sort of susceptible individuals being born, because immunity that existed previously is going away, because the virus is changing antigenically, because in some reason you don't generate to sterilize immunity, but herd immunity that is still contributes to infection. You don't get sick. And there's examples for all of these, and the population turnover hypothesis, sort of basically the birth of naive individuals, is something that happened for measles before we had the vaccine, right? Measles, quote unquote, strategies essentially gone, and generate around the community and rare reinfection patterns. is to some extent similar, right? Because they don't have as long as a lifespan as we. They don't get exposed to the same virus, especially domestic animals don't get exposed to the same virus multiple times through their lifespan. While sort of SARS-CoV-2 and influenza kind of sit on the other end of the spectrum here, where we have frequent or at least, common reinfection of adults, and the circulation of these viruses is mostly maintained by a combination of waning immunity and antigenic evolution, sort of generating some sort of approximate equilibrium between generation of immunity and its waning / change of the virus to escape that immunity. And this happens through viral evolution. And since this is so people using PDFs later sort of maybe we can figure out how the full screen mode works, that you can go slide by slide rather than having to scroll through a continuous document. But this is just sort of one example here of this antigenic evolution at the genotypic level, where we have the H3N2 influenza, which emerged in 1968. And I've colored this here. So you'll see quite a few next strain screenshots here, not because this is very specific to next strain, but that just because it's the easiest way for me to get these things. So my talk is going to be very, very little about next strain in particular, but you'll see a lot of it for mostly convenience reasons. So you see this here colored by the genotype at position 189 in the HA1 site, which is a position that sort of happens to change in a fairly regular and then also very sort of discrete fashion. So you've seen this sort of cycling through various amino acids over the past 5 decades. And this not just happens at position 89, it happens at dozens of positions across the genome, which I've some selection of which I've plotted here. This is just sort of diversity and alignment from the last decade or two. And you see that there are specific spots in the HA, mostly in the HA1 subunit, where you have a lot of diversity, and many of these sites map to the globular head domain here, where sort of specific characterized epitopes are. Maybe Derek will talk a little bit more about that tomorrow, or in the antigenic consequences of these mutations. But yeah, so the turnover, the genetic change at these positions results in antigenic change, which ultimately sustains the evolution and the circulation of the virus. And to basically respond to that, we have a well-oiled machine, GISRIS, sort of a surveillance program that's been running for 70 years now, I believe, that basically, at least now, sort of convenes 2 meetings a year, sort of often in Geneva, sometimes somewhere else, where mostly the WJ collaborating centers decide on the composition, sort of what strains, what representatives go into the vaccine. This is currently either 3 or 4 viral strains, sort of for the H1s, the H3s, and 1 or 2 of the Bs, where the B Yamagata is probably gone, so we don't need to put the vaccine anymore. So we're moving back here to a trivalent vaccine. And the input data to that decision is, for one, sort of the epidemiological patterns, the genetic diversity and the dynamics that has been observed in the preceding months and years, antigenic characterizations of quite a large number of viruses using ferret serology. And here, sort of, it's an important or an interesting aspect that before we were able to sequence these viruses, this ferret serology was basically the way to tell them apart and say whether they're similar or not. Now we kind of use their genetic sequences, their genomes, to tell them apart in a much more discrete and information-rich manner. But there has been there's a shift undergoing from kind of antigenic characterization first to sequencing first. And one can argue sort of how many identical viruses one needs to characterize antigenically in order to. It's a good way of introducing redundancy in the process at this point. But one shouldn't really expect a lot of differences if the viruses are identical. Then there is human serological data, often sort of comparing pre and post vaccination of vaccinees. And there is sort of, for the last 10, 15 years now, also sort of modeling and prediction approaches going into this, and outcomes sort of a vaccine composition recommendation that the manufacturers then implement a few months later. All right, so what kind of data, especially genetic diversity, are we then looking at? So this is sorry. This is a phylogenetics tree, sort of focusing on data from the last 2 years here, and it's colored by the broad scale geographic region as to where these samples are from. And what I think the main take home here is that most major clades have a bit of a sort of confetti of colors, meaning they are present globally in all major regions. There's a few exceptions to that, most notably this one here, which is China during the Chinese isolation, when nobody could go there or leave without sort of long quarantine, right? That is sort of the one instance where you have extended circulation of a variant in a geographic region that didn't mix with anybody else. This variant was not competitive outside of China, and it happens to be the only thing that was in China. Nothing else got in because of a very extensive quarantine regime. Otherwise, it's rare that sometimes you have little clusters where that are sort of very specific to a particular region, but largely, successful viruses everywhere in different proportions, but it's making its way around. The other thing that is very common is we see a lot of parallel adaptation, and we've always seen that sort of with SARS. Now this is kind of it's more well appreciated, but this is just one recent example here where different H3 clades have picked up the mutations at position 140, either to a K or to an M in different parts of the world. It tried multiple times. Ultimately, this big one here sort of took over. And to facilitate communication, discussion, and generally sort of getting on top of that diversity, these WHO collaborating centers and the GISRI system has for the longest time been calling these groups clades. And the nomenclature has usually consisted of number letter dot number letter dot number letter dot, sort of resulting in somewhat longer strings, which at some point then sort of get ad hoc shortened because one really only cares about the last little bit. And one thing that I can point out already, I'll come back to that in a minute. So basically, for about a year now, everything is this group up here, the 2A, 3A, 1s, and we've kind of lost discriminatory power of this nomenclature because everything's the same. So the purpose of this nomenclature is in many ways, and that's probably something that we're going to discuss for different viruses and in different contexts. We want sort of units, discrete units to track and predict what's going on, right? So you can't do predictions on the basis of an individual virus. We also know for sure that flu is going to be around next year, so that's not very informative. So we need some sort of meaningful breakdown of diversity in order to make meaningful predictions. And this allows us to track and also predict and extrapolate. Then we want to aggregate phenotypes in some meaningful way, right? So typically we think that phenotypes sort of change in discrete fashion across the tree. So, individual measurements are often noisy. So we want to aggregate them in some way that we get, that we put order into those data. Some people also use them for reassortment analysis. I haven't talked about this yet, but flu has 8 segments that reassort, with different rates in different settings. And, by having sort of a hierarchical clade nomenclature for different segments, you can look at different constellations and combinations. And then lastly, this kind of nomenclature is used for communication. For example, you tell vaccine manufacturers that the vaccine is supposed to be of that clade and that clade and that clade. And there's to various degrees sort of attached phenotypic meaning attached to these names, whether they are antigenically similar in some way or another. All right, so what, some examples. So this is just sort of tracking, this is H1 in this case, tracking of frequencies across different regions. You see some regions have a pretty good, data situation where the frequency estimates are fairly tight, and others it's very noisy. Here we have an example of the aggregation of the serological data, where ferret antigenic titers here raced, Sarah raced against different different viral isolates from different subclades here, are tested against lots of viruses, and these viruses come from different clades, so you can aggregate them in meaningful ways. And for example, see here, let's see C16 and C17 are not well recognized by this particular serum. Right, so just as an example, sort of the individual data points here are not too important for this purpose. And then this is an example of, what is this? Go away. Yeah, so this, it also doesn't help that Zoom is delayed. Because what I see here on Zoom is about a half a second delayed, so which generates unfortunate feedback cycles into this. I should look more. Yeah, so this is an example of a reassortment analysis that Becky Konder from the US CDC has started using, where you have the neuraminidase subclades and HA subclades, and you can track what combinations are seen and how that changes. Now, what are the challenges with this existing nomenclature? First and foremost, it's often just, getting it updated is very slow, because somebody needs to take the initiative, push this through, get everybody's consent, and then and then sort of a year gets passed and we're still stuck with the old thing. And this leads to, groups sort of inventing their own sort of little sort of ad hoc classifications, which sometimes works for a little while, but sort of gets, and people don't end up knowing what they're talking about to each other because everybody uses different things. The other problem is that these names get longer and longer, and this ad hoc shortening sort of results in ambiguities. And there's also currently no way to look up what the actual nomenclature is. So it's all some sort of you look in last year's report to see how CDC did it, and then so there's no sort of central source of truth, which is also something that I would say is a severe shortcoming of this year. Okay, one example of how things aren't up to date enough. This is influenza B Victoria, where basically since 2022 everything is V1A 3A2, which so everything's the same. That we might just as well call it influenza B, right? And so then, sort of how would one organize it in a slightly different way, and what else do we use it for? So influenza A in particular has been the subject of various efforts to predict the evolution. This currently sort of Marta Luksha, Dennis Roshnevich, Michael Lessig, and various people from NextTrain are active in this. They've used, historically, there have been a few other groups there. There's also now a few other groups. But essentially, what you want to do is, given this current diversity, you'd want to put your money on one of these groups that you think are most likely to dominate the upcoming season, right? And there's various different features that people use to do that. So one is sort of measures of phylogenetic expansion and growth. So this is one of the colorings here that I put. So basically this group has been growing quickly, and these subgroups here seem to be also growing quickly. Then there's various scores that you can build on mutations in particular epitopes, often sort of around the receptor binding site. Maybe we'll hear more about that from Derek tomorrow as well. Sort of mutational load, to what extent we think these viruses are loaded with deleterious mutations and hence more or less competitive. Parallel evolution is often a good indicator. If something happens multiple times across the tree, there's usually a good reason for it to happen. If something is quite distant from other things, there might be a reason to assume that it's antigenically distinct as well. Also, past circulation patterns. All these things sort of somehow feed into these predictive models. And I think many of these features will also be useful criteria for building some sort of score or algorithm to find the clades to begin with. And sort of what we are currently working on with the influenza community, so it's a bit work in progress. We haven't sort of fully worked out all the details yet. But the idea is to adopt this somewhat more sort of Pango style, and we'll hear more about from the creators tomorrow or later today, I don't know. Which is basically sort of a letter number pattern with letters kind of being unique shorthands for whatever was there before. And the clades are going to be sort of what we're currently settling on, is having them suggested by something that's vaguely automized, but then sort of picked on inspection, because we're not looking at thousands. So, and for the one of the things that I've always felt is important in this context is that one wants to add things to an already labeled tree. So when the tree is kind of growing, and at some point you decide, okay, this branch is going to get a new label. So it's a slightly different problem from de novo classifying an unlabeled tree. But I think Angie might also talk a little bit more about these challenges tomorrow. So what we've settled on for here is that we have some score of phylogenetic expansion. We have some score that in kind of rewards branches with important amino acids. There is something that sort of measures divergence from the parents, and then it's a minimal size criterion. They're combined, and then we are sort of trying to update this about 4 times a year, sort of aligned with the WHO schedule, where we have a GitHub organization that sort of maintains machine and human readable definitions. And we so far opted for this aliasing not being kind of always at a particular point in automatic, but something that we kind of decide more in a consensus manner when we feel like it's relevant. So this is sort of one example here of this, which is retrospectively, and you see that, for example, in this case, we basically jump from one to another. Retrospective is always easy. Basically because there's a lot of important stuff going on here, so we just give it a new alias right away. But other than that, so this one here sort of dragged on a little longer, and at some point decide, okay, it's almost fixed. To get now. So this is how this is currently working in terms of frequencies. But I think that is basically everything I wanted to say about flu. And how much time do you have? 50. Okay, I have plenty of time. Okay, I wanted to finish within a few minutes just talking about RSV, where sort of a similar kind of rethinking has been going on. They're actually a little bit further ahead with their redefinition of the nomenclature. But the status quo ante here was that there was multiple previous proposals, not so recently, that was tried to do a unified version, but by the time they were published, they both lacked a mechanism to update it in a dynamic fashion. By the time they were published, there were like 1, 2, 3 clades left, and essentially they made a post hoc classification, but nothing that sort of really is sort of dynamic and alive. So now, what do you do about it? There's a real risk that you end up in a situation like this. I guess everybody knows this little XKCD, right? That's ridiculous. There's 14 standards. Let's make one to rule them all. And then there's 15. But I think in this case, and this is work led by Stephanie Goya, I'm hoping that this will actually work the way it's intended. So this is what the classification currently looks like for influenza, for RSVB. This is both implemented in Nextblade and Usher. And you see here there's sort of the yellow, the different tones of yellow are a little bit hard to distinguish, but you get the general idea. So there is this consortium proposal. Stephanie, who I think is in the virtual audience, I was hoping to see her here. She has really led that and sort of got everybody on board and made it happen. So it's a fairly broad consortium. There's its preprint that is up. It is sort of a vaguely Pango style nomenclature, with one quirk that the aliasing here is cumulative, which was decided by popular vote. But we'll see how long these. We don't expect this to grow very fast, so let's see how that's going. But I think generally having a nomenclature that has it here is going to prove very very helpful. To the end, I just wanted to end with some few sort of general thoughts, which maybe can feed into various discussions that we have over the next 2 days. So that these publications are fine to announce criteria and systems, but they're basically completely useless for updating, because it's always just going to be too slow for something that is a rapidly evolving virus. And if you haven't built in sort of a way of updating your nomenclature, it's basically going to be irrelevant by the time you publish it. And so I think that's really what needs to be in the publication, right? Not the classification itself. Then another thing that having sort of a consensus nomenclature is probably much more important than having the best possible. And you require some sort of dynamic source of truth that's ideally run by sort of a broader community is probably necessary. And GitHub has worked pretty well here for SARS or Mpox. SARS being a much more dynamic entity than Mpox, but Mpox also following a similar pattern. The definitions and representative sequences should ideally be open and ideally be able to link to. So this has been can be a bit of a challenge because you need to, for all these tools that you want to build to assign sequences to lineages, you need to have access to those data, and in a way that's not necessarily behind various barriers. It doesn't mean that all data necessarily has to be open, but at least sort of representative sequences, I think, are something that we should aim for to be open. The challenges that I have faced in various contexts here, sort of balancing the proliferation of names with sort of a sufficient granularity to track. And what you want here, I think, really depends on what you're going to use it for. So whether you really want to track every individual little lineages, or whether it's you're kind of trying to capture epidemiologically relevant sort of large-scale dynamical patterns. Yeah, balancing aliasing with preserving a hierarchical relationship is also something that is sort of a difficult compromise to strike, right? So, if you want to preserve a lot of hierarchy, you'll get not long names, right? Or low granularity. There just doesn't seem to be a free lunch here. But it's something that, again, I think the community has to, a different community can do it in different ways. So this is, basically the competing objectives, tracking every emerging variant versus sort of labeling phenotypically different groups. Pronounceability is something, and memorability is also important, in particular when these things are supposed to be used in internationally diverse context, because it is surprisingly hard for some people to pronounce something like this. For others, might be easy. And what is hard is hard for different people. So one should have that in mind, that it's not run it by a few people with different linguistic backgrounds as to whether this actually works, right? Because I've seen people struggle with these things, and it's something worth keeping in mind. And the last thing is that, yes, having easy to use tools, I think is important. And if the nomenclature is implemented in an easy to use tool and it very easily becomes a consensus nomenclature, right? So having these tools at hand and these definitions up and openly available and all that helps a lot to get these systems universally accepted. And with that, I'll sort of acknowledge the relevant people. So yes, the WHO Collaborating Centers for Influenza for continued collaborations for many years. The various scientists and laboratories that share these viral sequence data, be it via INSDC or GISAID, Stephanie and the RSV Nomenclature Consortium, and of course the nomenclature team, and in particular Cornelius, who sits there, who has been the main person implementing the various nomenclatures in Nextglade and Nextglade, maintaining Nextglade. So forth. Also Ivan, who is the main software developer behind Nextglade, and you'll hear more about that from Cornelius down here.
thank you very much for the invitation to participate in this. And like everybody, I've got passionate views about nomenclature and have in the HIV field for many years. But my talk, Elliot asked me to speak about the biology and what's driving the evolution of HIV and SARS-CoV-2. And I'm going to try to give you a comparative exploration of how these viruses are evolving, and HIV is really different. So basically, something that I do for the NIH is every other Monday give a breakdown of what the new variants look like and what should be on the lab bench. So my reason to be is to try to figure out what's the form of spike that needs to be on the immunologist lab bench and on the virologist lab bench. And if you look historically, these are three different maps of the history of the SARS-CoV-2 virus and the subtypes and the variant forms. And you can see they really make dramatic shifts. And this can often be led by just a few bases or a few amino acids, or sometimes by many. And so what I've got on these three different maps are the counts of sequences that were obtained over time. And this is really the full period of the pandemic. And then you can see the number of cases below, which really peaked with the variants. So that's, a very important epidemiological parameter. And then we stopped tracking the number of cases in March 2023 because reporting was too difficult to track at any point. And I'm going to talk about these transitions in the talk. So the major shifts when you get a global sweep and a transition to a new form can be driven by gradual accumulation, where you just have a few amino acids changing, and I focus on spike. And I think that's really been the driving force for most of these shifts. So an example of that is going from the blue BA5 to the yellow BQ1. There's just a couple of changes added on top of BA5, and there was a global sweep. Recombination can lead to one of these things. So an example there is XBB. It's two BA2 lineages that had both diverged a little bit. They came together, brought some amino acids together, and then there were a few things that piled on those, a little bit of accumulation, and you got an expansion of the XBB lineages. And then you can have a radically different new form, at least radical for SARS-CoV-2, where there's a whole lot of changes. And these are probably arising in a single individual, although we don't know who that individual is, and seeded a population with a very effective virus. And I'm going to talk a little bit about how those might be working. And then you can get accumulation on top of those. And a good example of that is Omicron. And that's the purple to pink. There's just this incredibly rapid shift from Delta to Omicron. Just a couple months and the whole world had Omicron, and there was a massive increase in infections. So you can see how these things look on a tree. These are 100 randomly selected sequences per month. And this shows you kind of an image of what you get. You start at the bottom of the tree in the yellow, and that's the ancestral Wuhan coming out. And then you get a big jump to Delta and to Alpha. Alpha's the orange down there, and the group from Oxford had the realization that this might have been coming from a single person's infection. So Andrew Rambo pointed that out initially with his colleagues, and that's probably true whenever you have these major jumps. One of the things I wanted to point out here is how recombination muddles the tree. So if you look at the very pink guy up at the top, XBC 1.6, that's a BA2 Delta recombinant, and it's got this very long branch length. So that's not by year. That's coming out because recombination has taken a few bits of Delta, popped it in a BA2-like backbone. That gives it a really long branch length. So that distorts timing and phylogenetics, and people have to be wary about that. The other kind of artifact you can get with recombination is the XBB, illustrated by XBB. So that's recombinant between two BA2 strains. And you can see that kind of a turquoise color and a little bit of brown at the base. That's XBB. So the other kind of artifact recombination gives, it gives two shorter branches sometimes. It comes in between two divergent strains. So it can either give way out too long or way back close to the base when it shouldn't be. And that really impacts phylogenetics. I just want to really briefly touch on the SARS-CoV-2 nomenclature, and we just heard about that a little bit, but to have everybody on the same page, because we don't all study this virus, it's a very convenient and beautiful nomenclature, I think. So what happens is when you start getting 4 fields, B1.1.529, they don't add more fields on that for the shorthand name that people can pronounce and say, which is quite useful. So now this BA1 comes off of that 4 point designation, which is phylogenetically based. And then you keep going until you get 4 more, and then you get a new set of letters. And I must say, I find this, when I talk to virologists and immunologists, they find this extremely confusing. So we're balancing the simplicity of being able to say BC1 with the fact that people don't remember that is a B.1.1.529. And what I love about it is you can have the short name to say, but you can also have the long name to retrace the phylogeny. So we use those long names all the time, and I will in this talk. The other kind of key point is the preface indicates that it's known to be a recombinant. I think there are quite a few recombinants we've missed, but at any rate, or the field has missed, I should say. But they're very useful also. So you can see when you've got a recombinant lineage because it starts with an X. But then once this name gets too complicated, it reverts to the two-letter nomenclature again. So HJ is a recombinant from XBB, but it's got that shorthand name so we can pronounce it and talk about it. Okay, so I'm going to then talk a little bit about what's going on recently. And this is the kind of thing I do for SAVE. And actually, I've been doing since 2020 on calls that David Montefiore organized before SAVE started. So we used to have a call every Tuesday, and immunologists from all over the world would be on that call. And now SAVE does this for us, and Derek is part of that group, and it's a really wonderful group. So we as bioinformaticians talk to the virologist and the immunologist and try to feed them useful stuff for their lab bench, basically. And what the JN1 looks like is it's this very diverse form that then picked up a single mutation spike 455S. And it's that form that took off. So the deep blue band is the original clade. And then the JN1, the bright blue, is the one that really went global. And we've started labeling a few of the different clades within that. And those are the three colors at the top, but they're all JN1s. The point I wanted to make with this is when you break it down by continent, you can see that there are different competing forms prior to JN1 arising. So there was really complex and very, very different backgrounds in the different continents. It wasn't all one thing. And sometimes we forget that because North America and Europe contribute so many of the sequences that we have this dominant view. But really very different things are happening in South America and Africa, and we're losing that view because people aren't still able to contribute. So I think it would be really good to try to build up those resources and keep the sequences flowing in. But you can see that the colors are really different, and that just means that we had different variants in those different continents. So this is provided by Cornelius, and this is just a godsend because I find the nomenclature really complex, and this enables someone like me to know what's new in the nomenclature. And so these are now all of the different JN sublineages that have been named by Pango. There's a lot more variants. This is the tip of the iceberg, but these are the ones that they felt to be dominant and coming out. So I can use this to understand the nomenclature. And all of the colors that you see there are recurrent mutations. So they're coming up in the tree. These are sort of organized by the phylogeny. And you can see that pink bit is one of them. The red one is 456L that was also coming up prior to JN1. And you can see they're coming up over and over. This is these trees are pretty fragile because they're only based on a couple of base changes. And there's a lot of recurrence. So the literal truth of the tree is something that people need to be cautious about believing. However, they're still useful organizationally. Okay, so now I'm going to talk about how I pick up. This is just for people who want to look at this slide later and know what's in it, this complicated slide here. But one of the things I do is I take out the last 60 days of sequences and I look at the change in frequency 2 months ago versus 1 month ago and the frequency of sampling and if that's increasing. So I pull out all of the forms that are increasing, and those are Pango names on the left, although often these forms are in many different Pango groups. This is just the main Pango group. And you can see that many of these are recurrent. The ones that are bright yellow are the ones that are pretty frequent, and the ones on the left and the ones that are bright yellow on the right are the ones that are increasing at the fastest pace. And so the 456L aren't quite as frequent, but they're increasing faster than any other forms right now. And you can see that these are the number of times they've occurred within the tree, and those are reflected in the Pango nomenclature. So these are the most highly recurrent mutations that are showing up in JN1, and now they're showing up as doubles. And so those double mutations are now increasing faster. So when we're talking to the immunologist, we say, well, you might want to try the double one on your lab bench. And then we give them the exact form that's the most common version of that virus. So they all have known functional attributes, and thanks in part to the SAFE project, because we get stuff to people very quickly and they can test them very rapidly. But for example, we have R346T. We know that this enhances stability while maintaining an interpolymer downstate, which is interesting because all of these beautiful antibody sites in the RBD are accessible when the spike is open, but it's getting to be selected to be down, and that hides the ACE2 site as well. So this is a little baffling conundrum. So all of these have different functional attributes, but resistance is a big lead for selection. And you can see how these are coming up across all of the different pango lineages that carry these. So again, multiple lineages carry them, but you can see that these are three very important mutations. And the one that's increasing the fastest is 456L. It's up now about 18% of the JN1s. And then you can see that the combinations are coming up, and we can do this at continent, global, or country scale. And every place the combination of these two mutations is present is coming up the fastest. And these are the 6 countries where it's currently present. So we make this kind of summary, and now we've begun tracking convergence, and I'm hoping to have these tools available pretty soon at our Lano website. But what we do is we want to find the mutations that are established and beginning to spread. So we had to come up with a cutoff. We did this very arbitrarily, but if I observe something in a lineage at least 100 times, I think it's beginning to move out, and I start to believe it. If I observe it at least 10 times, it seems to be maybe at least transmitted in a local outbreak and viable. Could be a sequencing error, but a good chance of being viable. So once we know these two things, we can start tracking. We can say these are relevant strains and anything that meets this criteria, and we start to track it. So I have 3 criteria for positive selection: highly recurrent mutations, mutations that happen just once but give rise to a dominant sublineage. So something like the 455 mutation in JN1 happened way at the beginning and then really occupies almost all of JN1, but it didn't happen multiple times. It just happened a couple of times. And then sites that take on multiple amino acid changes is also a good indication of positive selection. We have additional, once I map all of these, I can then link it to Jesse Bloom's gorgeous data. So he takes these viruses and he studies them on the lab bench, and he mutates all of the common amino acid in every position and spike off a backbone, and then does phenotypic tests on them. So he can do things like look at serological escape for XBB 1.5. He's got that data. He can look at how a mutation affects viral entry and how a mutation affects ACE2 binding. So he has these 3 measures. So I wound up with something that looks like this. It's a spreadsheet where I have Jesse's data on the right. It's gray for all of these single mutations. I have a number of times they recur, and I have something I call parent and child lineages. I'll explain that in a minute, but I think the recurrence is somewhere between those 2 numbers. So in the XBB 1.5 lineages, this top most recurrent form is 456L. And then I have the number of times it's found within the lineage as the orange band. So these are evidence of positive selection. This is what I mean by parent and child lineage. So the Pango nomenclature gives us a handle on this, but we can see that there were 5 different lineages which gave rise to that mutation, and it came into 8 different lineage names. So I think that this recurrence happened between 5 and 8 times. And in the discussion with Angie, my inclination is to think that 8 is a better estimate of the recurrence, and she kind of felt that was her hunch as well. But I'll show you a little data for that. So the other thing we do is we keep track of all the places this occurs. It can occur within one lineage, or it can occur when a Pango lineage name change happens. And so I'm going to show you that in a second, what that looks like. So for example, this is the A475V change. The lineages start out with it being the A ancestral form, and then they go on to acquire the V, and you can see the rate of change in each of these lineages. And this is what they typically look like. So you can see these switches within lineages happening over and over in a positively selected site. So now we can then compare this to Jesse's data. So this is our mapping of Jesse's data, which he makes publicly available on his website. So you can see these were the strongest escape mutants that he had listed. They aren't necessarily matching up with the best candidates for positive selection. So they may have other attributes that make them limited in vivo setting. I think the top is what we're mapping. These are things that were recurrent and very common and had at least 100 representatives in the recurrent lineages. So these are the sites that XBB 1.5 liked and picked up over and over again and then really expanded. And this is how they relate to the phenotypic data. And when you look at XBB 1.5, it turned out to be really quite interesting. So the bottom now is just that I've gone from Jesse's extremely complex thing where he's tested all these amino acids at all these sites, every color is different amino acid, to just the amino acids that were under positive selection in XBB 1.5. And the ones in the RBD that are around 400-ish, you can see tended to be strong escape mutants. So they caused escape from neutralizing antibodies in Jesse's assay. But we also had some of our selected sites turned out to actually be more sensitive to neutralization. And yet they were selected and moving through the lineage. So we thought that was kind of an interesting counterpoint. So if you look at all of the things Jesse measures, this was very strange. The ones that are more neutralization sensitive bind ACE2 more tightly. So maybe that's a virtue for these viruses and helps them. But they had reduced spike-mediated entry. So this was an odd set of combination of things, and these were the selected sites within this. And I think understanding these phenotypes for these highly selected sites and what the virus is finding in vivo is going to be quite interesting. So the next thing I wanted to talk about applying this kind of method to is foreshadowing. And so the idea here is if you want to look at a transition between viruses and you want to say what's going to be selected and you want to anticipate what's coming. If you look at the field of mutations that were recurrent before that new variant comes out, what was getting positively selected over and over again? So it's getting selected once here, once here, once here, and then they all start coming together on the new form that's going to become the new global suite. And we definitely see that. So those are the mutations in alpha, for example, on black, throughout all of spike. And the bright blue ones underneath it were evidence of positive selection. So the two black ones on the end were not positively selected pre-alpha, but all of the blue ones were. So you can also see this thing happening with Delta. The black ones are all of the changes in the Delta backbone. Delta swept the world. And all of these things are positively selected in the pre-Delta world. So it was a pileup of these positively selected mutations that we saw happening one at a time. Delta gets a lot of them. And then Delta really moves out. And finally, you can see BA1 also had this kind of attribute. So the things that were happening in Delta, these positive selected mutations piled up. And that was the thing that became Omicron. So you can also maybe use this to compare viruses. And BA2.86, which eventually became JN1, wound up having a lot of changes over BA2, its parental lineage. And so all of those red bits in the middle band are what makes BA2.86 different from BA2, a lot of changes in spike. The ones that are labeled in blue were positively selected in the year prior to the occurrence of this virus. And so again, there was a pileup. 19 of those mutations were positively selected one at a time, two at a time in the viruses that were rising before that. The ones that are getting selected within JN1 now are the ones in yellow. They were also positively selected prior to the occurrence of JN1. And finally, BA87.1 was one that we all got really interested in because it was such a different virus, but it never went anywhere. So it arose about the same time. BA2.86 was in South Africa. BA2.71 was co-circulating in South Africa. But this one we only sampled 9 times instead of having it take over the world. And it had some positively selected mutations, but only about half as many as JN1 had. JN1 had 20, it had about 10. So these things are co-competing, and these are all advantageous sites that are piling up. You could have made a guess, although retrospective is easy. And I'm looking at it retrospectively. I would have predicted that JN1 would have been really highly advantageous, whereas the 871 not so much. I'm going to take you through really quickly what one highly recurrent mutation looks like. I think it's the most interesting mutation in Omicron because of its high level of recurrence. It takes on all these forms, but it wants to get rid of the R. So this is BA1, and these are all the Penguin lineage names. And all the gray ones have the ancestral form, but all of the other Penguin lineages that are pink and blue have taken on a mutation. And so this is what you see on this slide. It's the timing of this. So BA1 starts out with ancestral, and then many, many lineages acquire a K at 346. And 346 changes. So all of the BA1s that persist out to the end of the BA1 epidemic have taken on this K, not quite all of them, but almost all of them. And then BA2 comes in and wipes out the BA1 pandemic. But this BA1 with the 346K was the one that caused the huge spike that we had when Omicron was introduced in the world. So it was as if it kind of supercharged it a little bit. It's the one that came up in the spike, and that's what you can see on the bottom because that's relative to case numbers. And BA2 is the black, and then that's the switch to BA2. Okay, so same thing for BA2. It starts out with the ancestral form. It takes on all of these different kinds of mutations. And XBB is actually a BA2 lineage member because it's two BA2s that have recombined. And so what you can see is that these are the different forms of 346K that are taken by all of these different lineages, highly, highly recurrent mutation. And you can see that the blues are different forms of R346T that were very common coming up in the BA2 pandemic. And then JN1 comes along and it's a reset. We go back to ancestral. But you can see that the BA2 also shifted. You can see here's now JN1, it's beginning to pick up 346, but we're just at the beginning and this is just a few months. Okay, here's BA4. BA4, the things that persisted to the end of the BA4 part of the epidemic all carried a mutation. BA5, same thing, it was shifting and then the XBBs came up. So this site is recurrent and it seems to be kind of a supercharger and really shifting every single lineage that picks it up persists, and the ones that don't have it go away. Finally, this is a recombinant that also has this phenomenon where R346 changes. This one was really only present in Australia and New Zealand. It's the pink bit up there, XBC 1.6 recombinant. And again, it changes to pick up this mutation. So this is what recurrence does when it's a really highly positively selected site. And it means that I think we should be really interested in this site when it shows up in JN1, because it's probably going to be dominant. Okay, really quickly to tell you how different HIV is. So in 2000, HIV database hosted a group of experts to come in and say, how should we name these viruses? And we came up with this kind of system where we had groups, and groups were different introductions from gorilla or chimps into humans. The only one that expanded globally was the M group, but we do have a couple of other groups, and we gave those letter designations. We had subtypes that were really distinctive, and those get a letter designation as well. So these are all M group subtypes. Then we have subtypes. We break down that letter. So we might have subtype A and then 2 different subtypes, 6 different subtypes within that based on really distinctive phylogenetic lineages. And then we have something we call circulating recombinant forms. And when we held this meeting, this is what we had. This is sort of a group of representative viruses. And you can see on the left, we have these major clades within the M group, and they all were uniform and persistent between different parts of the genome. So there's GAG, POL, and that branch links are quite different. And then we have these 4 circulating recombinant forms, and they can take on different bits from different viruses. It doesn't just, there's not just, one breakpoint in these, there are multiple breakpoints often. So basically, Brian Foley gave me the slide. We now have more subtypes. So we have A1 through 6, and we have these little designations. We're completely saturating the silent transitions, and so those are not useful in a tree anymore. They just kind of add noise. And you can see where the M group down the bottom red sits relative to sort of the SIV world and the viruses that are in monkeys, which are much, much more diverse. This is now NextGen has this beautiful ability to work with the HIV database. And this is a pretty clean set, but this is 4,000 some viruses, and it shows you these major clades. And then a couple of the major forms that are CRFs, the CRF 07 and 08 are pretty much only in Asia, China primarily. So somebody I think needs to turn off their mic. Who is listening? This is what we do for CRFs, and this is at the HIV database. But now instead of 4, we have 157 different CRFs we asked that they be circulating. And if you click on those little buttons to the left, you can get a map like this that shows all of the different breakpoints where they persist and the different clades that they come from. So this is what CRF07 looks like and whether it's B or C clade contributing. And this is just, I've shown you this tree before that Bill Fisher gave me, but it's taking, 100 sequences per month for the whole SARS-CoV-2 epidemic. And you can look at the different clades, and I've labeled just a couple of them on the left. And you can also label this by time. And you can see that it kind of makes sense that as you're moving out in the tree, you're getting older, although you do have these recombinant jumbles a little bit. It's pretty sensible. If you do the same thing for HIV, it's chaos. And the reason is massive recombination. And this is something I think our field kind of doesn't, it's like, we don't like to talk about it because that means all of our phylogenetics is screwed, but it's there. So you can see on the left there's subtypes and CRFs, and this is the same kind of sampling, but rather than 4 years of the pandemic, it's 40 years since we realized HIV was in the world. And you can see that the clades are now this muddle. And if you just do the random sampling rather than this carefully curated, here are the major clades and here are the major circulating recombinant forms, if you just pick them at random over time, you get the by year slide over there is just really a chaotic mess. And you get all sorts of the recombination artifacts that I was talking about. With HIV, it's really hard to detect intraclade recombinants. It's an explosive star-like phylogeny. Part of that, I think, is because of all the recombination that's happening. So you can detect inter-subtype recombinants because you have these distinctive parental clades. Within a subtype, you don't know who the parent was. All you have is the child. And there's really no way to tell, but I'm sure that the intraclade is littered with recombinants as well. There are some attempts in the literature to look at that, but I think it's a very difficult problem. This is just to give you a sense of the scale of variation. We published a kind of a review article a couple of years ago comparing SARS-CoV-2 and HIV evolution. So it's sort of an interesting comparison still, I think, if anybody wants to take a look at it. But that top left is envelope, and it's envelope relative to a global consensus. And these are amino acid changes that are colored underneath. And this is what SARS-CoV-2 looked like at the time. There was, if you did a random sampling of SARS-CoV-2, you could sort of see alpha coming up, and then those were the other major clades that the WHO had labeled on the bottom. So that's all of spike, and those are all of the changes we saw in the first, year and a half of the pandemic, almost all white because there's hardly any changes one at a time. Whereas HIV is co-circulating mad diversity, and we're trying to build a vaccine. Wish us luck. But anyway, now this is what the most recent current selective sweep looks like. It's JN1. So all of those bands are about 8,000 JN1 sequences, and all those bands are where they differ from Wuhan. But even this is like nothing compared to the HIV diversity. And this is actually now a map of the most recent JN1 sample. So this is essentially selective sweep. Those are those recurrent mutations we're talking about. The world has reset. It's reset to JN1. There's almost no diversity in JN1. It's slowly building, amino acid by amino acid, pingles on it. But, we don't know what's going to happen. Probably either one of these is going to have a combination, becomes really advantageous. There'll be another sweep, or some other very diverse form will come in and have advantageous mutations, and there will be a sweep that way. That's at least what we've seen so far. So they're just really different world, that top left versus this, about what's co-circulating right now in the world. And I just want to make one other point. HIV continues to diverge genetically and antigenetically. So if you look within a subtype, you can see that the branch lengths are longer over time, even though we have this kind of muddling of recombination. The red are recent samples. This is by my colleague, Elena Georgi. And you can see that that sort of the pairwise distances are increasing over time. And those pairwise distances increasing have antigenic consequences. So this is, this is the starburst kind of phylogeny within a clade. And you can see that the blue, which are the recent isolates that have been tested for neutralizing antibodies, are tending to be more resistant, which means that the bars are a little bit higher. So over time, over a 5 or 10 year period, you see this gradual increase in resistance, and our field is really worried about this because most of our reagents, most of our pseudoviruses for testing are from, 1990s to early 2000s, really not past 2008. And these blue guys are from probably about a 5-year span, about starting at 2017. So we don't have anything really recent. We're not sampling it. There's nothing from 2024 to even look at. That was the C clade I was showing you, and this is a slide made by my colleague Shedid. But these are looking at a whole bunch of antibodies over time. And so red are sort of our current panels that we're using that are older viruses. Blue is the new viruses. And when you see this shift up, it means you need to add more antibodies to lots of viruses to get neutralization. So the problem is getting steeper and more difficult as we wait sort of at this 5 or 10 scale as we are working on trying to get a vaccine. And we're not keeping up with our reagents very well, although we're trying to mitigate that and get more new pseudoviruses in. The other thing that's changing is the global distribution. And the viruses, the subtypes and the CRFs are really expanding apart to the world that had been primarily one subtype infection. So you can see Europe over there, it used to be pretty much B clade, and now it's B clade and C clade. CRF1 has gone from really just being Southeast Asia to major parts of Asia and also Australia. And there's been shifts in Central Africa, and the virus is beginning to have different clades also present in South America. So we almost always, for our studies, our clinical trials are done almost always with B clade populations, primarily green, or Southern Africa, primarily C clade. But we call these global panels. They're not global. That's our vocabulary. A global panel would be representative of the world. All of Russia and the Middle East tends to be this A6 form. And there are really distinctive patterns in these clades in terms of antigenicity. So it's very important, I think, to have better sampling and more contemporary sampling. So in conclusion, highly recurrent mutations that give rise to stable lineages cause phenotypic changes, and they can be mapped. The driving forces are neutralizing antibody immune evasion, greater stability of the spike and enhanced entry or ACE2 binding, and not necessarily both at once. And I don't quite understand what that means, but that's what I'm seeing in the literature. Prior convergence foreshadows mutations that are concentrated in newly emergent variants that achieve global dominance. Mutations that confer immune evasion often had reduced ACE2 binding. So that was really interesting. And my colleague Priyambada Acharya has done some beautiful structural studies that are now showing that this 3D down conformation is really being selected in Omicron, which obscures the antibodies, but also obscures the ACE2 binding site. So that's a mystery yet to be solved, how the virus is coping and compensating for that. HIV-1 is vastly more diverse than SARS-CoV-2. I think that's primarily because it's a chronic infection. And there's this cat and mouse game of get infected, get antibodies, escape, get antibodies, escape. In every single HIV infected person, this is driving radical divergence in envelope. Influenza, SARS-CoV-2 doesn't have it. Hepatitis C does have a similar kind of situation. It doesn't have the recombination muddling things, but it really, I don't think there's a one size fits all solution to nomenclature, I guess. So that's it.
So this is our first Q&A panel discussion. And that suggests that multiple things are going to happen, and that's what we're trying to opt for. That is, both will take questions, and we'll start with questions both from our in-person audience and from our Zoom audience, and we'll be relaying the questions from the Zoom audience. So if you're remote and you have a question, please type it into the Zoom Q&A feature, and then we'll have someone here read that question to our panel. And then after we've taken questions, then we have a series of questions that we've defined for the purpose of the workshop. And those questions are really made to lay out some of the important points that we want to discuss over the 3 days of the workshop. And they're meant to guide the overall outcomes of the workshop. And so let's just get started. I think we do have one Zoom question. Indresh, would you like to read the one question that we have?
Yes, this question.
And please turn on your microphone.
Yes, it is on. This question comes from Rumini Gao. and this is for this came during Richard's Neher presentation. For the IVA reassortment analysis, what are the criteria to define the genome where the reassortment occurred?
Well, in this particular case, the hope is that one doesn't actually need to define these criteria too closely, right? Because you classify the tree of both the HA and the NA segment, and then look at what combinations of these clades do you observe and how that changes over time. So essentially, you've kind of outsourced the need to detect the reassortant to detecting combinations of clades of NA and HA. But this is actually early days. So Becky Condor and her team at US CDC were just starting to use this way, and we'll have to explore more how this is going to work in the future.
All right. Do we have questions from people here sitting in the room? Yeah, Derek.
Thank you. This is for Bette. I'm thinking about the 346 substitution, and you very nicely pointed out how that seems to be, not only supercharging, but also sort of different from other substitutions. We're used to many substitutions getting fixed and then carrying on as lineages proceed. Do you think there might be something about 346 that it might end up causing a lineage to go extinct, so that when then some other lineage comes up without it and then needs to acquire it again? I'm thinking in particular about the hypothesis of it keeping the spike in the RBD in the down configuration, and whether or not that's some evolutionary dead end for the virus.
That's a great question, and it is a little bit baffling to me. I mean, one of the things about it, though, is in South Africa, when they first discovered and presented BA1 and BA2 simultaneously, essentially in South Africa, they had the ancestral form, and then 4 and 5 also had the ancestral form. So the seeds for many of the Omicron lineages came from that. And those lineages, so that might have been kind of driving what we were seeing. It was really favored, but all of the baseline that spread globally had that ancestor. However, Nicole Doria-Rose on our last call had a really interesting suggestion trying to understand this. And her thought was perhaps in long-term infection, you have a tendency to select away from the R346 plus a mutation. So it doesn't survive well in a long-term infection. So when you have something like JN1 come up, it wouldn't want to have the variant there. Whereas when it's spreading at the population level, the resistance that it confers enables spread and blocking transmission. So I thought that was an interesting hypothesis, but it's a hypothesis. I don't know. But yeah, it's very puzzling. And I think the other really interesting sites are the flip mutations that we all got so interested in XBB, the 455 and 456. That 456 is kind of doing the same thing that 346 is doing. I didn't have time to, do a second series, but, and it came up later. So it came up in XBB 1.5, but meanwhile, in a lot of the other BA2 lineages, it was also coming up, and the 455 was also coming up. So before JN1 hit, everything that was persisting tended really frequently to carry those two mutations. And they really hadn't been sampled a lot prior to sort of the last half of the Omicron. And now they're getting resampled, and now they're showing up again in Jan 1. So another thing that suggests maybe is that there's time for particular mutations, that maybe our population immunological profile has sort of shifted about which antibodies are seen well. So something tends to get more selected based on the way we've evolved our immune response. And that's another hypothesis that might explain what we're seeing. And it's also really interesting to me. So the 346K was what came up with BA1. The only time we'd really seen a 346 mutation much before that was Mu. Do you guys remember Mu? Okay. So it was it was a really interesting little subtype that Alpha was kind of taking over the world slowly, depending on what it was competing with. But then Mu shows up in South America and Central America and is moving a lot faster than Alpha before Delta hit. And it had 346K also. It was the only place it was there. And that seemed to really be moving that particular lineage along. So it could be that K was advantageous earlier. We made antibodies that saw the K. K is not useful anymore, so it goes to T. So that's another hypothesis. Why was everything getting reselected on K initially, and then everything gets reselected on T? Could be that we've shifted the pressure.
Related to that, Bette, to what extent, for different viruses, and certainly SARS-CoV-2 and HIV are impacted by founder virus bottlenecks, but how might that be a more general phenomena that impacts the subspecies evolution?
So with HIV, I don't think we've seen these kinds of bottlenecks actually so much. We found HIV after it had been evolving for maybe 70 years in people. So it was really diverse when we first saw it. We didn't recognize it right away because it was an immunodeficiency disease. So it didn't cause one disease. It killed us by many methods. It killed us by sarcoma, it killed us by pneumonias, it killed us by intestinal diseases. And so we attributed them to something else because it was opportunistic infections. So it took us many decades to spot HIV. Once we finally spotted it and realized it was immunodeficiency, it evolved these clades already. They probably already evolved in Central Africa. So those initial clades you see are very old, and there was no bottlenecking. It doesn't ever get less diverse. It just keeps expanding, and it keeps expanding within clades, and it keeps shuffling between clades. So we have no bottleneck in HIV. And I think that is a very different way to think about nomenclature. And I would say the same for HCV. So, I don't think there is a one solution fits all. I don't think we should have a recipe for that. I think we need to look at the biology and the evolutionary biology of the virus we're dealing with and think of logical systems based on what we've learned. And we've learned so much from SARS-CoV-2 and how useful nomenclature's been.
And that's certainly a topic that we're going to address over and over again. Let me see. Other questions? Yeah, Richard Sharman.
Hi, Richard Sharman, National Library of Medicine. So, Richard, you made an interesting point about, from a nomenclature perspective, that we have to balance this proliferation of names with having sufficient granularity, and there's definitely a balance there. And Bette, in your talk, seeing the HIV community seem to have come to the conclusion that having kind of broader clades was sufficient for the purposes of tracking the evolution of HIV, which is very different from the nomenclature we have from SARS-CoV-2. So is there a happy medium there somewhere? I mean, how are we going to make a decision about how to strike the right balance?
Yeah, that's a tough one. But I think it ultimately, it will depend on what you end up using these things for, right? Sort of in SARS, we want to label all these lineages as basically as early as they are recognized, and then that's what's happening, and I guess we'll learn more about that from others. For flu, I think the community is happy to wait until something has sort of some sort of 5-10% frequency and a few mutations that we've seen before to give it a name, something that's sort of worth talking about, something where we expect we have more experience in what kind of mutations generate what sort of phenotypic signatures, right? So just seeing a mutation, there's one particular combination of mutations, sort of 145, 189 in the H3s that might mean something. We've seen it, in the sequences we have, maybe 50, 60 times dispersed across the tree, right? But any of these individual ones, no real reason to give a name at this point, right? But at some point, if we see one of them grow, we'll give it a name.
So now that we're kind of transitioning from pandemic to endemic with SARS-CoV-2, maybe we need to change our philosophy and adopt more of a flu-like philosophy. I don't know.
I think there's a couple of issues there, but one is the robustness of the clades, and because of all of the recurrence and the fact that the clade or a new name is often given to something with maybe one or two changes, that it's not really necessarily a stable lineage. And having done phylogenetics for for decades, we had all these tools to say, how robust is this branch and how robust is this clade? None of those tools work. However, that said, I find the names really important for testing psychologically. It's really interesting to me. So trying to get my immunology colleagues to test something until they've seen it tweeted, they don't want to test it. And so we're kind of in a different social world now. And it's an important thing to understand. So names are really important. And they make, so I can say, these mutations, look at these mutations. And they say, and then they say, have you seen this tweet? And then they want me to give them that particular pango lineage, which I always try to accommodate. So I think we need to think about that a little because it's a real phenomenon. And I think anybody who's been in our SAVE group has probably seen that too. Yeah.
But a quick follow-up on that. So robustness is actually crucial, right? For flu, typical flu segment is 5% of the length of the SARS-CoV-2 genome, right? So that that severely limits how much you can, how granular you can chop this up, right? In particular, if you don't want to go with sort of a random synonymous mutation to label something, and the branches are quite unstable at times, and the new data comes in, and suddenly the things get switched around. So that's why you can't really do the same thing for flu that we have for SARS.
The other thing is we don't really name every base, or we base. I think the ones that are interesting mutations and that are expanding are the ones that tend to be named, or interesting mutations, even if they're small. So if something's only observed a handful of times, but it has a cluster of interesting mutations, at least we'll hear more. But that's my observation is those are the things that get named, because any one of these pango lineages, once it's sat around for a little while, it doesn't always get divided, but there's many, many forms. So each one of these is a complex array. It is a clade within itself that's evolving. And there's millions of different variants just in spike. And we've only got thousands of pango names. So we don't name everything, but we might try to hold off until something is really expanding. And I think this is actually practically what's being done, like that one new virus that I was talking about in South Africa that was so different. There's only 9 samples. Tulio in South Africa pointed out we have this really interesting variant. It's super interesting. It is moving in South Africa. It's an important one to understand because it's so different and to get a handle on early just in case it expands. But it isn't expanding. So just expansion isn't a good enough criteria. And just differences alone, maybe not either. So some kind of happy medium between expansion and do we believe these mutations are interesting for some reason? And then give a name because no one will study it till you've given the name. And I do believe that's true.
Garrett?
Thank you, Elliot. It's a quick follow-on from this. I think another difference between flu and SARS-2 is that with SARS-2, there were so many sequences so quickly. And before we had Usher, for example, just new trees would come out every once in a while, that we got used to not mostly looking in the trees and associating what we were doing in the trees for most of us. And so I think then having a whole bunch of pango names and following substitutions from them, that really helped when we didn't have the trees. And Richard, in Fluland, yeah, it's true that we don't have names for all the clades, but we get to look at all those clades, right? Because there's not that many sequences. And you, for example, with next strain, for example, you make these great trees that people can use. So you don't necessarily have to have the name when you can look at the whole ancestry there, because it can be seen so clearly. So I wonder if that is a difference. And it's not so much that SARS-2 needs to go into Fluland. It's more that I think it depends whether or not you do have a tree where you can be looking at versus, or you're used to looking at. I know that we now, thanks to Usher, we do have trees that we can look at routinely, versus whether or not we've got to do it in a more non-tree way. I know that you're using all the tree stuff, Bette, when you do your great stuff, but a lot of times we're using those pango names to sort of abbreviate the tree structure.
Franca for the biologists, I think, the Bengal lineage names. You have to talk to them. You have to be able to talk in that language.
And please give your name and affiliation when you ask your question.
Please speak in the mic.
Okay, so because there's so many sequences, having somewhat granular lineages helps a lot with finding your way around the tree when you look at it. And as Bette says, she looks at the number of independent occurrences of mutations to as an indicator of whether something might be selected for. That's something that if you don't give something a name, it's much harder to count these things. You can of course write an algorithm and go through the Usher tree, but that requires a lot more tooling than simply counting, how many lineages are there. And yes,
Richard,
right, which isn't happening, because if you look at the Pango designation GitHub issue, there are some people who are tracking all the 90 or more than 90 346T sublineages of JN1, right? They don't all get names. It's really, maybe there are 10 or 15, the ones that are sort of where we know that, they're in several countries. There are kind of a couple of 100,000 cases around the world of this. But it's a good point that, there's a question, how many lineages do you need? And I suppose, having many sequences also allows you to do that. And as we get fewer sequences, there will be definitely fewer lineages.
I guess another way of looking at it is that, one can use these, especially the hierarchical nomenclature system, as a substitute for phylogenetics, right? So we are basically setting up a whole bunch of buckets in which we can sort the sequences into. And, depending on how many sequences we have, that also determines how many buckets we need to put things in, right? And whether it's worth setting up that extra bucket.
And another point is it's not just sort of numbers of sequences expanding globally, but our sampling is so poor in most of the world. So we have these really rich sequence sources from a couple of countries and parts of Europe and a few countries in Asia and North America. And then, so this means if there's only 30 sequences coming out of Brazil in the last 3 months, but they all look different, 30 is a big number. Then if it's 30 out of 30, that's different. So we should care about that small number of sequences in a poorly sampled place if it's looking interesting.
Should remind everybody, please use the microphone when you can. And to our AV guy, there are a number of microphones that aren't working. Apparently, a whole row of microphones is not working. Okay, we're going to take a question from Zoom, and then we'll come back in person.
All right. So there are 3 questions from Zoom. I'm going to read first one, and then
Indresh, can you speak in the mic? We can't hear you.
this is working, I guess. First question is for the talk RSV, flu, and the beyond. Are the suggestions for how we select lineage for names for automated suggestions, and what people or group of people should be responsible for? Something between a small study group or frantic free of the SC2.
So if I understood this correctly, is sort of what are the criteria to designate a new lineage in RSV, and who makes a call? And so there are in this preprint that Stephanie and the consortium put out, there are sort of a few criteria there. There are some suggested algorithms, for example, Autolin, that Angie will talk about tomorrow, that basically makes these criteria quantitative and concrete. But I think probably at the end of the day, there will always be a bit of sort of a human curation step. Whether this is consortium, a study group, or just people that are engaged and end up putting it out there. I don't know. I don't think we have sort of, well, there is a consortium in a way, but I don't think there's a formalized decision process at this point. But if Stephanie is online, she can maybe put it in the chat.
Okay, let's take one more from Zoom, then we'll come back in the room.
Second question is from the Deb Wedford. Are these non-spike mutations that are show similar positive selection over the time, or that show convergence akin to D614G or R346?
I didn't quite understand the question, but are you asking about mutations not in spike that are also very important? I think there are some. I don't usually track them very closely, but some of the things I've done in the past is looked at sin non-sin rates, and there's the tendency outside of spike is that the ratio of synonymous mutations is much higher, which indicates it's not usually under such positive selection, but any given mutation, of course, might be important. It doesn't mean that they're not, but within spike, the non-synonymous to synonymous ratio is just massively higher throughout the pandemic. So I think most of the positive selection is focused on spike. That said, obviously there can be really important mutations outside of it, and I don't tend to look at them. So others might be able to address that better than I can.
Hi, I'm Rachel, and from the University of Edinburgh. Question for Bette, please. You showed very nicely the huge difference between the diversity of HIV and of SARS-CoV-2, and how something like the Pango system for SARS-CoV-2 could never really be applied to HIV. What's your feeling for how well the current nomenclature is for how good the current nomenclature is for HIV, or whether you would, in an ideal world, design it differently, working in that field now?
Well, so when we had that meeting in 2000, we set the nomenclature ourselves. It was within the community. It wasn't given to an international body. And we did it at the database, but it was, I think, because of these star-like phylogenies within each clade, and I think it actually is a pretty serviceable nomenclature. It's not an antigenic definition necessarily, although whole clades will miss a whole class of antibodies, which is kind of interesting. And we forget that. So, for example, V2 antibodies, which are a really hot target for HIV vaccines, work pretty poorly in the B clade, US and South America and Europe. They're great for South Africa. And another example is CRF01 completely is silent for V3 antibodies. So there is some clade associations, but then within the clade, there will be a whole spectrum of reactivity. And I think that the nomenclature to serve the immunology community is part of what we need to do. We need to give them a language that enables them to do the experiments that will tell us if we've got a working vaccine, if we need to change things, what we need to test. And I think that the HIV nomenclature does a pretty good job of that, except for we tend to just focus on B and C clade, which is a problem. They're going to, I believe, have a Zoom nomenclature call at the database. We discuss the HIV nomenclature. And so sometimes you get subclades within a clade that are pretty well defined. Like we went from just A to A1 through 6 over the 20 years since that nomenclature was instituted. And when do you make a decision like that? We actually had defined really carefully criteria about when to do that. And there's not very many new clades, but the CRFs are really exploding, and I'm not sure how useful they are. So we have 157 CRFs, so these circulating recombinant forms. And our criteria for circulation in 2020 was that it was found in 3 different individuals. So that meant it was moving through the population. If these breakpoints are in 3 people, okay, it's moving. So there are some laboratories that have made a career of finding circulating recombinant forms, and every time they find 3, they get a publication, and when it's published, it goes into our database. And there's other countries that don't care. So like Brazil, we have tons of BF and BC recombinants. They're not named. So we just call them BF, BC. Probably some of them would be a circulating recombinant form, but we don't have an attentive, biologist getting the CRF paper out, because you usually get a paper when you set a new one. So it really depends what country you're from and what laboratory you're from, whether they're named or not. And they're thinking about maybe making a higher standard. So you have to have 10 or 20 before you can call a CRF rather than just 3. Most of them are that site that I showed you that Brian Foley maintains. He helps people do that. And we have all kinds of tools for defining intersubtype recombinants in the database, make it very easy, make pictures really easy. And the number of times they're found globally is written there and where they're found in all citations. So it's a very nice tool if you're interested in seeing that. But I think that's the main problem with our nomenclature, actually. I think our nomenclature fits the phylogeny pretty well. And it is not necessarily an antigenic map, but it really does have a lot to do with the immune system because certain classes of antibodies are pretty useless in certain clades. And that's important to know. These are targets for vaccines and immunotherapies.
This is Ryan from NCBI, Ryan Connor. For Bette, it's sort of 2 parts, but for the HIV nomenclature, how do you feel about not the approach to naming things, but just literally the names you give things? If it was more the Pango style or is like letter dot number dot number dot. I mean, I imagine it wouldn't go super far out in the case of HIV. But like, do you see that being a problem in terms of just trying to standardize how the things are named across viruses? And then the second part is, do you feel like there's less of a push to get more granular, like subspecies classifications, partly because for the existing available therapeutics, the drug resistant profile is pretty well characterized, so you don't need to rely on the phylogeny as a heuristic?
Those are both really good questions. I think the robustness of the tree is really a problem, and I don't trust the phylogenies very much because of all of the recombination. So I don't think they're a true evolutionary history of the tree. Really, and so as an overarching grouping, I think they're useful, and as a sort of a rough tracing of the evolution, they're useful. So the kind of granularity that you can get with influenza or SARS-CoV-2 is not really, I don't think, meaningful in HIV. I don't think that's particularly helpful. So I don't, we're not actually getting these sequences in very much with HIV. And I think maybe this is also something we should discuss. So the history of HIV has really shaped the way a lot of you guys work. And I don't know how much you know this, but in 1990, we had contaminants in HIV in a major science paper, and there was no journal that required submission to GenBank. GenBank existed, but you didn't have to put sequences in GenBank if you published. And there was a massive letter writing campaign saying, when you publish sequences for transparency and reproducibility, you have to submit to GenBank. And it started happening, and it happened in all the major journals, and it was wonderful. And for years, we kept that up, and the editors and the reviewers kept that up. And now I think it's really fallen to the wayside. So people are not necessarily submitting. No one's calling them on it. There's many more sequences. It's much harder to submit, and you've got to have the metadata to make the sequence useful. So you need to know where it's from when it was isolated. And that's a lot of work. And if no one says, you have to put this in GenBank, people often don't. And we've really let that slide. And I think maybe a reinvigoration of the notion of when you publish the sequences and you give them to the community. And there's IRB issues with that, but to the extent that you can outside of IRB issues and particular studies, I think it should again become mandatory. So that's one thing. The other thing is there didn't used to be dates in GenBank. And the HIV community gave you guys that. So back in the 1990s at the database, we would read every paper and put in the date if it was there for every single sequence. And in 2000, I did my first timing estimate with the phylogeny. And then a lot of you guys have, I think some of you in this room have worked on that since then. But we had to really argue with GenBank at the time because they didn't want to have a field for dates. And in HIV, we can't put in the whole date because there is a wonderful project officer named Bonnie Matheson who was worried about patient privacy and HIV. It's so sensitive. She was afraid that if you said you're from the city on the state, someone could realize that you were HIV positive. So we were only allowed to put in years. GenBank often has specific days of sampling, but at our database, we only have years, and that's historically why. And that changed around the year, late 1990s, around 2000. So anyway, that's just a bit of history, but I think we need to rethink being reviewers and editors, requiring submission of sequences, and really being strong about it, not just letting it slide.
Over here.
Thank you. Yeah, Anya O'Toole, the University of Edinburgh. So my question is also for Bette. So I thought it was quite interesting you presenting about foreshadowing for the signatures of positive selection and the sites that we now kind of know as the defining sites of these variants. And I wanted to ask about whether you've looked beyond the sites that we now know have been, swept the world. Do you see signatures of positive selection quite often? Like, how permissive is that? So my question, I guess, is around how, if you don't look at the sites that you've confirmed or have swept, do you see positive selection in many other sites, or is it actually quite a true signature of what we'll sweep?
So there's more sites that are positively selected than come up in the new sweeping variant. You saw XBB 1.5 because that's all I kind of had time to show, but we've been looking sort of pre all of the big sweeps. And so we go maybe that like Delta before Omicron, or pre-alpha, and then we take out any alpha and we say what was happening. And there's always a good number of positively selected sites. And the older, of course, the older the lineage becomes. So like BA2 is really old because it goes all the way to the present, right? BA5 is it had a long time in the world. So it got really diverse. And so we come up with maybe 60 or so sites that are under positive selection, and we can rank them according to how many times they recurred and began to spread with that cutoff. And then we can rank them about, okay, if it's early on and only happened once, but it took over the lineage, that's interesting too. So we have those two ways of ranking, and I couldn't show you all we've got some nice little XY plots where, you can see the positively selected ones, and you can see the lineage that comes next, and they're up in that high corner. So what happens is always you see recurrent sites. The recurrence is massively important for this virus. And they change over time. So I think maybe our immune pressures are changing over time. Maybe with vaccines, we're just, you start out and you've got a Delta infection, but then you get an Omicron infection, and then your population around you is also getting that. So your immune system has changed. So what's going to be positively selected is going to be evolving over time too. So we see shifts in the sites that are important over time, but we always see recurrent positive selection. And we always see in these newly emergent forms, we see a really high percentage of sites that are among those positively selected just prior. So that's what I mean by foreshadowing. And we're seeing it consistently every time we've looked so far anyway.
Let's take a Zoom question.
Charles, there is a large lineage inclination towards human viral pathogens. Shouldn't we also give importance to animal viruses due consideration? This is because similar classification challenges also exist for the public health.
Yeah, definitely, and I think this is happening for various pathogens, right? So we have an extensive nomenclature system for the H5 avian influenza viruses. I guess we'll learn a little bit more about the swine flu classification from Tavis later. So for the flu world, this is certainly something that is happening, and I'm not so familiar with other animal viral diseases, but I imagine that whenever it's appropriate, people do come up with these, and we're hopefully, going to be in a position where we can make this more systematic and sort of more powerful with better tools and better infrastructure in the future.
Yeah, I agree. And I think that the focus is where the money is, right? So that agriculturally impacting viruses are studied and classified to some extent. And, that I wish that extended further, because we've got a lot of species on the brink right now. But I think I think there is an interest when there's a agriculturally relevant virus, people do sequence it and try to work on vaccines and try to classify.
Another Zoom question, and we'll.
Sure. This question comes from Ellison from HHS. So really important that we maintain the ability to capture difference that impact medical countermeasures with the SARS-CoV-2 naming convention. I would think if you transition to something more similar to flu, that granularity would be lost and that it is very important for public health advances.
So if I understand that question correctly, the responsiveness of the SARS-CoV-2 is of value right now. And the fact that we're getting a lot of names when there's something that could be relevant is important because it helps us understand the evolution of this of the pandemics and the currently endemic variants. I agree with that. Actually, I don't think we should go to a very simple system quite yet. One, because I'm finding the pango lineage immensely useful myself, and I think that I can help translate it for the virologists who need to be studying these things on the bench still. I mean, we still need to know what's the vaccine for next year. And by having people looking so attentively at this, we can really nail down which ones need to be tested better. I think it gives us a framework. So I think it is helpful.
I have a very brief comment. I'm Elliot Gedden. I'm at NIID. And Bette, it was really interesting to hear the history of metadata and release of genomic data. But I think we're going to right now, we're hitting another problem in the metadata. Clinicians have been telling me that in their hospital, they're now being told they cannot release dates, especially in these cases where we're looking at emerging of lineages from these immunocompromised patients. And so now I was trying to get, month and year for this publication. They're saying, well, we can't put the year. I said, but we're talking about a lineage that we know is related to, the era of the lineage. And so there's going to be some pushback because with AI, the hospitals say you can now really track who the patient is.
That's a great point, and that is exactly what we confronted in HIV when NIAID said to us, "You can only put years in." Okay, if people want to put, because it's okay with ARRB, dates in GenBank, fine, but we don't want that for the HIV world. Only yearly was fine for HIV. It's not fine for this. And the other point, actually, you bring up a great point for nomenclature, I think. So Angie pointed me to a paper when we were talking on the phone about back that sort of has characteristics of longitudinal sequences. So you can maybe try through bioinformatics to guess what's longitudinal. But in fact, labeling single patient data, and if possible, given the IRB for a given study, the dates of sampling, and having that be part of the metadata very, very clearly is extremely helpful. And human readable, we can often in the HIV database say, this is the same patient. These 3,000 sequences are from one patient. We can know that kind of, but there's no systematic way of doing that, where it's very clear that this name goes with this individual. So we read the papers, we tag an individual, and then we link all of the sequences in our database. That's one of the things we do for the HIV community. Obviously, couldn't do that for SARS-CoV-2 because there's too many sequences. But if we had systematic ways of doing that across viruses in the metadata, I think it would be immensely useful. And I don't know why they won't let you say a year. That's strange.
Well, so now, we sort of have gone back and forth, and we feel we're threading, a needle to be able to not provide too much information. Although they're like, oh, it's fine if you put it in GenBank with the metadata. I said, well, it makes no sense because then somebody can actually track it.
Yeah.
But in the publication, we wanted to put January 2021. This is what's happening. They're like, yeah, you can't put, January. You can say 2021. But I think right now we're all trying to figure out how to do that.
How to handle that. Well, certainly in GISAID, which is what I use for my data sources, there's a lot of sequences, not a huge amount, but a lot of sequences that just have year or don't have a date. And that's probably part of the origin of that. And I think if you're constrained by IRB constraints and patient privacy to do that, you have to do that. That's the thing to do. But to the extent possible, to give dates is great for the larger community. And, I've been arguing this with people in HIV for years. And they don't, big IRB sometimes don't want the dates in. But I think if you ask the patient when you set up the IRB and you said, do you want the date to go with it? Your blood from your arm would be so much more useful to the world if it went with the date. I bet the patients a lot of times would say yes if someone asked them. But it's not always in the protocols for asking at the IRB of will you release this data? Let it be the patient's choice, maybe not and that's not in the culture.
So in a lot of the data that are, but relevant to that, the age, other metadata that could help identify the patient, like sex or age, I think is, so I don't know how much of that information is included in a lot of the metadata, or whether it's not, there's just date and no other patient information.
With HIV, it's all over the map.
Yeah.
And we just take what we can get and put it in. But I do think a systematic way to separate within patient studies and clear nomenclature for that would be extremely helpful in any viral field. And we don't have that right now. And every group just does it their own way. So that could be a useful thing out of this meeting.
So we have time for one more question. Go.
Sorry, it's Rachel again. It's more feeding into the conversation that was going on just there. The main thing that we've seen being involved in the COG UK and projects that have happened since has been with the date issue and privacy. It's more not the date itself, but how the date associates with other metadata. And we've resolved it, instead of always changing the date, we've sometimes resolved it by making geographic location more vague, changing the granularity of the location, or not giving the other metadata gives you many more options if the date is the thing that we really, really want to keep, which often it is. Like sometimes that matters even more than what scale of location you have.
Right. With SARS-CoV-2, the date really matters. With HIV, the year is really all you need. So also this is maybe a virus specific thing about what's useful. Yeah.
Okay, one last Zoom question. And
that's the last question I have. Question comes from an anonymous user. Is there or should be certain % of variation for Omicron in order to designate it as a different lineage? Are there any cutoffs at this time?
Here you go.
Well, I think sort of I understand the question that it is about how do Greek letters get assigned, right? And for intents and purposes, this has just stopped. This initially they were given out sort of very liberally, sort of when something was growing and we didn't really know much. Sort of with basically ubiquitous seropositivity and the concomitant sort of lower severity, there is very little reason to assume that a new variant that is going to behave fundamentally different is going to pop up. So I wouldn't expect there to be a pie anytime soon.
I think we're coming near the end of the hour.
Just picking up that point. Sorry, Peter Simmons here from Oxford University. I think the variant of concern nomenclature is actually quite useful, and maybe the lack of allocation there's been recently because obviously there haven't been variants of concern particularly that had the impact of the old ones. So, perhaps it is logical, and maybe you could tolerate a dual nomenclature for SARS-CoV-2.
Well, I was not so much saying that I don't think that, if one could have gone on and given out more Greek letters, right? It was just by whatever body gave those out, they decided that that they're not going to do it anymore because they're looking for sort of a severity signal or something like that, right? In terms of genetic diversity and distinctiveness and sort of also growth rate of novel variants, that would have, if those were the relevant criteria, we would have had more of these. But given that sort of severity was always sort of, or at least some fear of severity was a central criterion that stopped. But in terms of BA286, was growing very fast, was very, very different. It was bigger jumps than comparable jump to BA than something that could have been one.
Yeah, and JN1. Yeah.
All right. So thank you, everyone. Thank you, Bette and Richard, and the audience for a very stimulating conversation. I think all of these issues that we've talked about are going to be critical to our ability to come up with a consistent method that impacts all viruses and our response in the future.